All right, our next presenter is Katelyn Anderson, and she is in Kevin's lab.
Hi, I'm Katelyn Anderson. I'm in Dr. Strychar's lab, and we are identifying Escherichia coli and other bacteria at Hoffmeister Campground Beach using imaging flow cytometry. So, for those of you that don't know, Escherichia coli, otherwise known as E. coli, is a gram-negative bacteria that's present in the digestive tract of humans and many warm-blooded animals. And it's also present within the waters of the Great Lakes and surrounding inland lakes and rivers. And we monitor it because if it's in a high concentration, then it could make somebody sick if they were to ingest it. And it's also an indicator that there could be other pathogens in the water. So to start the research, we wanted to see if we could establish a live dead count of E. coli in the water. And we used this live dead kit by Thermo Fisher, and it came with cytometry and propylium iodide. And what it did is, if you see this, there's a bacterial cell right here. So the cyto9 stains the cell fluorescent green, and it will stain every single cell that is present within the sample. But this PI, on the other hand, stains the cell red. And when a cell fluoresces red, that means that it has a damaged cell membrane, so therefore we would consider it to be a dead cell. And then we later found out that cyto9 actually is not specific for E. coli. It stains 14 other different types of bacteria. So we found this other probe stain by integrated DNA technologies that should stain a cell that is only E. coli because it uses this DNA sequence right here. And what the company did is they linked this fluorescent dye to the 5' end. And from this picture here of an E. coli cell, you can also see that it also fluoresces green like the cyto9, but it is only E. coli because it contains that DNA sequence. So when we started this, we had several objectives. Like I said before, we wanted to establish a live dead protocol for E. coli, and we think we did that. We thought we did that with the cyto9 and PI, but we found some problems. And then we also wanted to see if we could move into specific species like swine, bovine, or avian. And we also wanted to create a method that is comparative to Maggie's Kohler and to the qPCR, because Kohler only measures the culturable cells in the sample, and qPCR does do a live dead based on DNA, but it takes several hours for both of them to come up with data. And we wanted to see if we could develop a stain that would identify E. coli versus any other bacteria, and we think we found that with that DNA probe. So to start this, we collected water from the north, center, and south ends of the Hoffmeister Campground Beach and used 1 liter bottles. And from that, I filtered it with a 5-micron filter into 100 mils. And then from that, I pulled 1 mil sample and put it to a 1.5 mil tube. For the cyto9 PI, I would use 100 microliters of sample, and then I would put 1.5 microliters of each dye, and then I incubate it in the dark for 15 minutes. And then once that was done, I would vortex it and then put it into the cytometer. And for this DNA probe, I would also use 100 microliters of water, but I would only put 0.5 microliters of that probe, and then I would incubate it in this thermomixer at 1,000 RPM for 30 minutes at 22 degrees Celsius. Once that was done, I would centrifuge it, and then when it was done centrifuging, I'd pull 90 microliters of that supernatant out and add 90 microliters of PBS. So this is our imaging flow cytometer, and flow cytometry has actually been around since the 1970s, and what happens is a cell is suspended within a liquid, and then it will pass through several lasers that are inside the cytometer, and if it's labeled with a fluorescent dye like the cyto9 or the PI, it will then emit light at a wavelength, and then it is picked up by a cytometer and is placed in these graphs right here, and you can then use those later for analysis. And what's different between a regular flow cytometer and the imaging one is that ours actually has a camera that is inside of it, and it takes individual pictures of each cell, and it makes analysis easier for when you're trying to figure out what the cell is. So this is a graph that was created by the IDEAS program, which is the analysis program of the cytometer. And this is based off of the intensity of channel 5, which is how bright the cells are that are stained by the propylium iodide. And it's also with the intensity of the cells that are stained by the cyto9 and how bright they are. So when I looked at this, I noticed there was 2 different major regions of cells that was being formed, and I figured that was probably from the different stains. And then there's also a smaller region right around here that when I first looked at this, I didn't really notice it, but after looking at it more, I determined that this is actually what the graph became. So these are images of cells from these regions. As you can see, the dead cells, this is the cell, and then it's stained by both of the dyes because cytometry stains both, and propylene iodide only stains one with the damaged membrane. And then the live cells, you can see it's only stained green, and there's no cell with the propylene iodide. And then that smaller region, by looking at each individual picture of them in that area, I could tell that this was a bead because it is smaller. And the beads actually are just something that run in the background and they help with analysis because they can be next to another cell and just tell you what the size is by looking at it. So this is the result table. And as you can see from the live dead concentrations, they were way high compared to the color. The color was getting about 10 to 20 CFUs, and we were getting several thousand up to about 3 million cells. And that's when we stepped back and we looked at our methods and we found out that the cytometry does not just stain E. coli, it's also staining other bacteria. And that's why we decided to find that DNA probe that would only stain E. coli cells. So this is that probe stained cell from through the IGS thing. And this one is also based on the intensity of channel 2 and the brightness of the cells when they are stained with that probe. And then it's just based on like the area of how big the cell is. So this one had some clumping of cells too in different areas, higher and lower in the graph. And when I looked at it further, I noticed that the top one was actually the protein cells and the bottom was the beads. And then if you look at this big region right here that I called inaccurate cells, this is what they looked like. We don't know exactly why they did that, but we think that it may have been centrifuging and it just created a big cluster of cells that were stained. And I put them as inaccurate because I wouldn't count them in the final count because they're a little big. But for the probe stained cells, they all kind of look like this or a little smaller, and they're all stained with the probe and weren't clumping. These were more beads that ran in the background, and then this cell is unstained, and all the cells in that area were because there is no DNA probe, so therefore it's not E. coli. So this is the results from running that test with the probe. And if you look at the concentrations of pretty much every single sample, they're pretty low compared to the cytometry PI. Yes, they're higher than the color. Color was pretty low still, but we figured it was more accurate, and we're still trying to figure out different ways to make this number more accurate to the color, and hopefully we'll figure out a way to do that soon. So for the future, we're hoping to get into source tracking, figure out which animals are contributing to the E. coli in the water. We want to get back into that live dead count to determine if the cell is alive or if it's dead, because if it's dead, it won't make somebody sick. And we eventually want to compare it with qPCR. Like I said before, qPCR takes a longer time, but it does give a live dead count. And we wanted to see if we could create a method that's faster, and hopefully we could get results in the same day. And then we're working on refining the methods right now, just kind of getting rid of some of the centrifuging, changing the incubation times, changing the temperature, just trying to make it more accurate and make it so everyone knows like that it's safe. And I'd just like to thank Dr. Strychar for teaching me about flow cytometry. And I'd just like to thank everybody else that taught me about beach monitoring. And I'd like to thank AWRI and the college for allowing me to do my internship here over the summer. This is my lecture. Are there any questions?
So I have one.
So what's your next steps to modify the methodology? What's your next plan?
Right now, I'm actually working on centrifuging versus not centrifuging. And then we're thinking of trying to different temperatures, like making it hot, making the sample hot as it's incubated or cooling it down. And then we're also trying to think of maybe changing the incubation time because all of those, like this probe could stain better or worse depending on, we're trying to just figure out better ways to do that. And right now the centrifuging, there's a little bit of a difference, but we're not really sure. Yeah, still working on that.
And then for the first stain, that was a little bit less specific to E. coli. Was that also a DNA-based stain?
No, this was just like a basic, I think it was like a nucleate stain.
Gotcha. Okay, so it just stained DNA in general.
Yeah, it stained, it would stain thousands of cells, whereas the DNA stays only a few, like maybe 20 or 30. Yes.
Any other questions? Great, well, thank